Unpopular decision to have less camera on the dog and more camera on me. So, good morning, good morning, good morning. I'm having some internet problems here this morning. I believe you're here with me. I hope you are. Okay, I'm now seeing the chat interrupt. Yes, Doug the dog definitely loved Riggs's cricket pup cake gift. It's, he inhaled it, as you saw. Hooray. Thank you for sharing that you can hear me. And welcome to Friday. I hope it's sunny where you are. It is sunny where I am sitting and standing. And so Doug the dog and I are happy to be here with you today. And we're going to talk about Lofaphorates. Lofaphorates, which is the group that you didn't know that you wanted to know more about. We started talking a little bit about the Bryozoans, these moss animals. They are animals, they are not moss. On Wednesday. And today we're going to continue talking about the Bryozoans. We're going to add to them the Faronid worms and the Brachiopods. Remember on Wednesday we talked about one of the enigmatic position that done it all in the 2014 paper from the Annual Review of Ecology and Systematics talked about regarding the Lofaphorates. Are they a monophyletic group or where fit the Bryozoa? And we presented kind of two hypotheses about that. I'll keep working the door. Welcome, welcome, welcome. Oh yes, you're very attentive this morning, aren't you, dog? Aren't you, dog? That is, in fact, the last cricket pupcake that I have. Now, before we get too much farther, I have a little thing to share, and that is, first of all, thank you for the coloring, and I will clear that. And Sherry's been posting jobs, USRA jobs, URA jobs, those kinds of things, they come up. This one came across my Twitter feed yesterday. So applications for Ontario Park summer positions are now open. There's a link that I'll post in the chat, the lecture chat at the end, along with all the videos and links to any of the primary literature that we're using in the text. And so you can go and explore that if you don't already have a job. The parks in Ontario represent some of the most amazing kind of landscapes and have a multi-tiered kind of responsibility to those landscapes, to welcome people there, to keep them clean and to protect them. And in some cases, if you're up in the Renfrew County Algonquin region, to extract resources from them. So it's a neat organization. It's a great way to spend your time. But we continue. Today we're going to be diving into chapter 17. There's three taxa, three phyla within the spiralians, within the protostome. So the Faronids, not very species rich, 18. Brachiopods, not currently very species rich, but they have been, in fact, during many eras in life, in geological time, life on the history of our planet. Brachiopods appeared to have dominated the marine environment. And then the more species rich, about 6,000 species of Bryozoan. And remember, we talked about where they sit. And so they've listed it here in this done it all phylogeny as an unresolved polytomy. And in Bruska et al., they actually talk about this. They refer to the fact that the Lofaphorates, the lophophore-bearing animals, have had a long and tortuous taxonomic history. I feel like your voice should warble as you read that. Come here. Come here. Can you sit again? Sit. Where's your paw? Where's your paw? That one. Where's this one? Where's that one? There you go. So, the first phyla that we're going to deal with are the Bryozoans that we already started talking about. So 5 to 6,000, they called moss animals. And what you're looking at here is a colonial collection of multiple Bryozoan zooids. This is the box in chapter 17, which remember, is a great place to go back to touch base and to refresh yourself from the lectures, to capture lots of the detail in terms of the apomorphies that characterize this group. So we're going to talk about a lot of these features, but generally they are sessile animals, sessile colonial animals that live in marine and freshwater habitats. Now, this is also a capture, again, from that done it all phylogeny, where we talk about, it's kind of a character matrix associated with the phylogeny. So it's a great pictorial way of capturing and representing what kind of characters each one of these taxa has. So we can look at gains and losses of some of these defining characters. So they have nerves in a basement membrane, they have a brain, They don't have ventral nerve cords, lateral or dorsal nerve cords. Bryozoans are, as you see here, with a through gut and anus mesoderm, lacking nephridial cells, so no kind of excretory organs, with a blood vascular system, but with no segmentation. Bryozoa. What does that mean? It means essentially moss animals. So when I think about bryozoans as a nerd from the 70s and 80s, I tend to think about the swamp thing. That essentially the swamp thing as a moss animal would have been characterized by some taxonomists as bryozoans. We have a rich, there are some aquatic bryozoans, freshwater bryozoans in and around the Great Lakes watershed, but there is not an enormous diversity. There was a great big diversity here. Stop. Many years ago, when Guelph was the bottom of a shallow tropical sea during the Silurian period. Now, where I grew up in the Ottawa Valley near Algonquin Park was on a little tiny strip, literal like kilometer wide, 20 kilometers long strip of Ordovician limestone. And one of the things that I tried to do this fall in preparing for our distance-based offering of 2700 was drive around and take camera equipment to outcrops of limestone rock in Devonian, Silurian, and Ordovician, so kind of bracketing just before and during the evolution of fishes, when a lot of Ontario was underneath warm tropical oceans, and when we had a large diversity of now principally marine phyla that we wouldn't see in Guelph necessarily. And so I'm going to give a shout out today to my parents, Clark and Elaine, who took that broom and they went out in December just before Christmas and swept off the exposed limestone of the Ordovician rock where I grew up, and they tried to take some pictures of some of the fossils that occur in that Ordovician limestone, including bryozoans and brachiopods, two of the phyla that we're going to talk about here. So I'm going to try and do something clever. So wish me luck. Tell me in the chat real quick if you just transferred from the screen of the PowerPoint to this screen of the interpipes. Thank you. Thank you for doing that also so quickly. So this is a little bit of that limestone rock, and if we zoom in to this particular fossil that looks, as I'm tracing it here, like a little, I don't know, kind of like a little teardrop that has these arms or surface areas radiating out here. This is a fossil, this is a sheared fossil bryozoan from the Ordovician period, now found in Renfrew County. These little tiny areas, this is actually where the zooids, where the individual animals would have lived, and they would have, this is if it's, kind of reverse it so that those zooids are pointing up, imagine, into the tropical ocean. And that lophophore, that tentacle with ciliated tentacle containing coelom of the organism would have been out there kind of bringing in food and particles from the Silurian shoreline into their mouth so that they could feed. And now they're captured in this rocky outcrop, and I will give, I will give a bonus mark of life if anyone can tell me what that not fossil is on the same rock outcrop. Enter it into the chat if you can see that. We'll zoom out. You can see some karst topography, the erosion of that limestone rock. Ah, moose poop. So, poop, yes. Moose, no. Moop, no. It is poop. Yes, that is in fact sheep poop. This is the only, it does in fact looks like poop. That is the only vertebrate poop that we're going to see in this course. And that is, of course, because my parents, actually this is another thing that's shared between Sherry and I, both of our parents raised, well, in my case sheep, and in her case goats. So let's escape out of that. So those Stop. Sit. So those gigapans, we may explore them later on as a way to jump into and try and identify some different life forms from the Devonian, Silurian, or Ordovician in that case period. So thanks, Mom and Dad, for taking some of these pictures. There's a whole bunch, there's like 6 or 7 phyla in this particular picture here, from gastropods to cephalopods to echinoderms, brachiopods, and bryozoans. So remember in that upside down tear-shaped little bit of bryozoan, there were those passages, those channels. There's a name for that. They're called zoocoea. And these are images, scanning electron micrograph images of individual zoocoea from extant or from living bryozoans. And you can see, so in each one of these little zoocoea, the little zooid of the bryozoan would live and extend the lophophore out of. And we watched some video of the lophophore on Wednesday. I think I've got some more in this video, in this presentation later on. But that's the little opening that they would have everted their lophophore through. So this makes zoocoea. Zoocoeum is the singular. That literally translates as animal house. And so that makes me think of the late departed John Belushi. And if you don't know the film Animal House and you're looking for some mental, some complete turning off of your brain this weekend, explore what 1978 brought to the world. It was more than just Dr. Jacobs, it was Animal House as well. So this is now looking, so we've got a zoocoelum here. This is an extant kind of a diagram from chapter 17 of your text, looking at the lophophore. Remember that cilia-ridden, coelom-bearing tentacle that's out into the water filtering. And we've got different structures here. So the coelom, where the antagonistic body walls, remember we talked about the hydrostatic skeleton before, they still exist here. So there are two different divisions of the coelom within the bryozoan. So we have the mesosome up here containing the lophophore with a tight sphincter that kind of divides it, and then the metacoelom or the metastome where the majority of the body occurs within that zoocoeum, that exoskeleton. Now, they're sessile. They have a U-shaped gut, which you can kind of trace in this little stained image here. And they secrete many casings or tubes. And there's another look at that quote that I like earlier on about the long and tortuous taxonomic history. This is another view of that body plan from, so it's comparing and contrasting. In this case, this is the diagram from the older text that I like because it brings out a couple of things that your current text and the comparative figure would be the last one we saw, 17.6. But you can see the mesocoel there extending up into the tentacles of the lophophore and the metacoel down here inside of the body. And then these connections, these funicular cords that connect the individual zooids to make essentially a colonial animal. So metamesocoel. So the zoocoeum being that individual casing, the exoskeleton. The coelomopores, which was a word you've seen in a couple, I've traced it there. I'm tracing it right now. The arrow's pointing at it. This is a ciliated groove in the mesothelium along which eggs move to be released out of the mouth into the environment for reproduction. The funiculus that I referred to, that's this there it is here. This ligament or tendon, connective tissue, let's call it, that connects the stomach to the bottom of the body wall and then allows essentially the colonial animal. Now, how do they feed? Is the lophophore out all the time? And it's no, in fact, as you probably imagine, as a clever and learned invertebrate zoologist. We need, again, firing of muscles across this hydrostatic skeleton. So in this case, we've got parietal muscles, which are around the center of the body. And the contraction of those parietal muscles, if you imagine kind of that I'm holding in my hands here, a water balloon. Okay, and the tie-off is up here, and the tie-off of that balloon represents the lophophore. If I fire those parietal muscles, that's going to stick up the tie-off of the balloon, and in fact, in the case of the bryozoan, it everts the lophophore. Now, the parietal muscles, so there you can see them firing, and the lophophore, so we scroll back. That's exactly what's happening there. So that firing of the parietal muscles across the hydrostat is averting that lophophore. And then the lophophore themselves, you can see in this video, this microscopic video, the beating of the cilia, creating water pressures, water currents, and feeding currents. And then the fact that they do have, remember the U-shaped gut, that was the bit we were sticking for till the end. So we talked about sheep poop. I have to balance that by talking about an invertebrate poop. So this is the Bryozoan in slow motion pooping. So the mouth and the anus right beside each other. The gut then descends and is protected inside of the zoocoelom. Working the door. Hello, everyone, and welcome. We're talking about lophophores, lophophorate animals, and in this case, Bryozoans. So physiologically and physically, the zooids are integrated. So the coelom is shared, and that their form depends on how we find them on exposed rock or exposed leaves. Depends on what sexual state that they're in. They're modular, but they can form through asexual budding. They can as well. The colony can grow. Now they also have, as we saw with many, maybe most, probably most beautifully, let's say, within some of the cnidarians, a lot of polymorphisms, where as a colonial animal, there's different jobs that are taken up by different parts of the organism. So in this case with the Bryozoan, you can see there are autozooids, and they're feeding. So they're doing kind of what the video that we saw a second ago, that was likely an autozooid. So it's just sending out the lophophore, building up a current, and bringing food into the mouth. There are heterozooids that are non-feeding zooids, and they have different kinds of jobs. So this first one is an Avacularia, which, so it looks literally like, I guess like some kind of a Muppet-based interpretation of a raptor, of the beak of a raptor. So that's used for defense. So they have zooids that are adapted towards defending against perhaps little browsing fish. And then they have vibracula, which are these cilia-like structures that keep the colony clean and sometimes can be used for locomotion. Because as you've seen in many cases, even when we say this particular taxon is sessile, what essentially we mean is mostly, maybe up to 99% of the times, but in fact some of these can move small amounts. Now we see different shapes that the colony assumes, and just like touch back to the sponges where we talked about we named sponges based on the forms that they assumed in the water column, and we talked very specifically about the fact that those names for sponges aren't phylogenetically based, they're describe the morphology they assume. And the same is true here. You can have a fructose colony that we described looking like a budding fruit body, or a fallacious, so it looks like a leaf, or in this case, literally, you can see where moss comes from. Or a stolonate, so it looks more like a hydroid, so branches and maybe more like a pine cone, a branch with pine needles branching off of it. Right, it's easy. So the encrusting form is the largest form. So you can see here the openings of the individual zoocoea, and then the autozooids, the feeding zooids, kind of branching out and batting and bringing food into their mouth. The Vivicularia and the Avacularia then kind of feeding and potentially helping create colonies around it, feeding motions around it. There's a quick shout out to the fact that those of you I'm going to show a video. It's not from, and it includes some BBC footage, so it is feeding, so we might have to trim it out of the posting on YouTube because of copyright, but we're going to watch it, and so if you're joining us asynchronously, make sure to get the links that are posted in the chat so that you can go and watch this later on.
What looks like moss or lichen is actually a tiny animal. Bryozoa colonize the kelp blades. Filtering water for microscopic bits of plankton. They hide from predators in tough shells that have a similar composition to those of crabs. But the Garibaldi fish's mouthparts are tough enough to rip the Bryozoa from the kelp, shell and all.
Boo to the fish. But what beautiful footage. So integration across the colony, and we've seen this video before, but here's a close-up look at that, the feeding autozooids. Now the integration, one of the ways, and this has been tested in some of the participants of years past who've gone on the marine and freshwater biology field course would have done things like this, where they stain different parts of a food and then apply the food in one part of the colony and then watch the colony kind of share that dye-rich food across. And you can see here in this particular example where the dye is absent, like in these cells here that I'm tracing with my little laser pointer. These are non-feeding zooids that were for either protection or for potentially for reproduction. Now, reproduction, lots of larval stages within bryozoans. They have what's called a siphonaut larva, which is right here. We're going to take a look at a scanning electron and SEM of them in a second. But these are long-lived for many months, and they're planktotrophic, and they have planktotrophic but also some lectotrophic. And so planktotrophic means they have a functioning gut, and they're actually out living for long periods of time and actively feeding. So there are, we talked about sessile bryozoans. That's referring, of course, to the adults. These larval stages, especially containing the planktotrophic larva, they're mobile. They're part of the plankton. Later stages, as they begin to develop, become lectotrophic, or later species, they can become, get out. They have a yolk, and they are feeding for a short period of time. Here's a look at what that siphonaut larva looks like. Now, the statoblast, just as we saw with sessile sponges producing gemmules, these can produce essentially little time-traveling and traveling in time and space, little packets of reproductive material that can be released out into the water column and allow them to live through periods of maybe desiccation, maybe ice if they're freshwater ones and living in our area, our part of the world, or just allowing them to travel long distances and to resettle and to colonize new places. So statoblasts, each colony then arriving from asexual budding from that first or zooid or an ancestrala. Now, bryozoans, again, if you're looking, as you go home maybe, or you're at home already and thinking about talking to your family about what you learned today, and they're like moss animals, and he said the swamp thing, and that looked pretty cool. Or it didn't, you know. Bryozoans can be used to produce cancer-curing drugs. This might be a way to get people more interested in thinking about Lophophores. Now, so bryostatin is a compound that's incredibly useful in not just cancer, but in HIV treatment, cancer immunotherapy, and the treatment of Alzheimer's. And there's only one source that we have currently in the world from a marine bryozoan that you're looking at in that video there, called Bugula nectarina. And it takes 14 tons of that bryozoan to extract 18 grams, so 3 elephants to make a salt shaker amount. So you can imagine that it's quite cost prohibitive, so an expensive drug. And it's actually not the bryozoan itself that's producing the bryostatin, it's a symbiotic bacteria in the gut. And in the wild, that compound is used by the bryozoan larva to protect itself from predators like fish, that Garibaldi fish we saw in the last video, for example. Now in 2017, so somewhat recently, we've been moving with success into with biochemical solutions that allow us to successfully synthesize this compound and maybe leave the 18 or the 14 tons of free-living bryozoan there where it belongs in the water, but still extract the good for human society in terms of eradication of HIV, cancer immunotherapy, and treatment of Alzheimer's. So if you see on the stock exchange, if you do that kind of thing, a pharmaceutical startup called Bryologics, that's referring to bryo, so you now know where the name on the stock exchange has been derived from. So that's a long time talking about bryozoans. We have 2 other groups to get through, but I want to make sure that I verify la chat and come over here. I'm a little bit confused. Bryozoans are within the Lofaphorates. Yeah, so Lophaphorata is potentially a group. That's one phylogenetic hypothesis that contains the bryozoans and the Faronids and the Brachiopods. So the organisms that possess a lophophore. There's an alternative hypothesis that the bryozoans independently derive the lophophore, so we talked about that on Wednesday. You can revisit that, and we'll talk about it a little bit more. How do they become colonial? Do they join together at a younger stage? Yes, that is exactly it. So settling down and then asexual reproduction, once the ancestral has arrived, both sexual and asexual reproduction. Okie dokie. So we're going to move from the bryozoans to the faronids, which have been called, in some of the places you'll see it called worms. They're not worms, they're worm-like. They're vermiform, which is a way of saying worm-like, but they are certainly not worms. And we'll talk about worms most of next week when we get into the annelids. So these are vermiform. There you see the word right there. Vermiform Lofaphorates, where the gut is U-shaped. The anus, just like we saw with the bryozoans, is close to the mouth. The marine benthic tube dwellers. And they're not so species rich. Right now we have about 18 species in the world. We talked about when I can bring up the derivation of words for some of these taxa, because in many cases it helps moss, animal, arm-footed. You're good. I will try and derive or tell you about the roots of a lot of these words as we go through these phyla. In this case, with the faronid worms, it doesn't really help you describe or capture any of the life history of the organism, because the faronid is from the Greek Faramis, who is apparently a mythical priestess of Argos who is loved by Zeus or Isis, and it's kind of uncertain. Etymologically uncertain as to why we've called the faronid worms Faronis. But there you take a look at one there. To me, it looks very much like a sock puppet, like a Muppet sock puppet with the lophophore around this mouth that has the hanging kind of protective cup now in this particular image here. And then you can see the full animal from figure 17.1 of your text with the lophophore out and the tentacles extended, and then a long trunk with the end bulb. And that end bulb for the faronids is important because it roots them. They essentially are a lophophore with a long, gorgeous, intricately arranged lophophore tentacular mouth, but their bulbs end planted into the sediments. And so they live in these permanent chitinous tubes where the lophophore is the only thing that extends up into the seawater. And there's lots of now increasingly molecular evidence that groups them very closely with the brachiopods. So as we talk about the enigmatic position of Lophophorata, what we're pretty certain about, we have a high degree of certainty in the close connection between the faronid worms and the brachiopods. It's the relationship between these two phyla and the bryozoans that it continues to be disputed. The hypothesis that we've presented so far is that they're likely the most parsimonious hypothesis is that the lophophore evolved just one time, and that they are, in fact, the three of them are closely related. So 14 to 19, 14 to 20, fewer than 100 species. They're not worms, marine sessile, and they have an internal asymmetric left side dominance, which you might have perceived in that line drawing from the Bruska et al. Text in one of the last slides. Now, the arrangement, the circular ring of the lophophore, there's a neat kind of a condition here where if the smaller you are, the less ornate and ringed and spiraled the lophophore is. So if you're very small, you have this kind of circular ring. As you get bigger and bigger, you develop a folding back, so that horseshoe shaped of the ring. And then as you get larger and larger, you develop some simple spirals in that horseshoe, and then complex spirals. And so, again, another application of an increase in surface area required to deal with the increasing volume associated with a larger body. Now, one of the cool things that, to me anyhow, about the faronids is that they have corpuscular hemoglobin and a hemosystem. So they're a very small animal. If you take a look, you can see that actually in the size. So in millimeters here we're talking. So body length in millimeters. This is not meters long, not centimeters long. These are very small, finger or smaller organisms. So it's kind of a surprise to see one of the apomorphies for the group being that they have this protein oxygen binding molecule in a hemosystem. So it's a very small animal. So why not just diffuse, as we've seen as an effective solution for most of the small animals that we've talked about here? One of the hypotheses is that the evolution of hemoglobin in this and a hemosystem in this small stalked marine worm is that it's an adaptation to living life in anoxic or hypoxic conditions. So why would they be anoxic or hypoxic? Why not just live where things are not like that? Well, it has to do with the fact that the lophophore extends up into oxygenated water. But this water is not, they don't have a tube to circulate oxygenated water to the rest of the body that's underneath the surface and contained in that tube. So perhaps a solution to living in this anoxic or hypoxic condition where part of your body is exposed to oxygenated water in a solution and part of it is not, is to evolve through trial and error over millions of years, a hemosystem that's very similar with high efficiencies that are very similar to our own. So there's issues with hemoglobin though, because extracellular hemoglobin, like we have, it's large. Intracellular hemoglobin is small. So at equal concentrations, small molecules of hemoglobin can bind more oxygen because they have a greater surface area. However, if you have a whole bunch of small molecules, you can increase the osmotic pressure. So small oxygen can combine more oxygen, but cellular hemoglobin helps you reduce the associated problem of having an increase in osmotic pressure. Now, how does that affect, or how does that pertain to the Faronids? Well, Faronids package small hemoglobins intracellular to enhance oxygen transport with high binding efficiency while minimizing the effect on pressure and on blood osmolarity. Now, oxygen transport, we haven't talked about this a lot yet in invertebrates, but we've in this class in the evolution of morphology and evolution, but this is in fact a kind of morphological adaptation to being an invertebrate. And so we can see oxygen transport like this with hemoglobin in actually a surprising array of invertebrates. We see them in nemerteins, in annelids, some mollusks, Faronids, Acardian worms, and echinoderms. But it's almost always extracellular. And so this is kind of hemoglobin, yes, but in one case, here with the Faronids, intracellular. Now I'm going to ask you a question about this. We're going to have a bit of a poll, a discussion and a poll based on some of these questions. So we have nucleated red blood cells circulating in body fluids in 5 invertebrate phyla. In the echinoderms, in the mollusks, in the annelids, in the Faronids, and the nemerteans. And so my poll question for you is going to be, if that's the case, and the slide here is going to stay as I set up the poll. When did the evolutionary adaptation of having nucleated red blood cells and a hemoglobin, when did it evolve? When did circulating nucleated red blood cells evolve? Based on this, so that's where the trait exists in these phyla, and based on this phylogenetic hypothesis, has it evolved 1 time, 2 times, 3 times? And I'll give you a second, and I'll deal with the dog who is chewing my jeans. Come on. That's good. I can see just about 2/3 of you have voted. I'll give it a little bit of time more, a little bit more time. If there's anyone else who would like to vote, we're going to resolve this hypothesis democratically, which is hardly ever how science works, but that's how we're going to do it today. Okay, 75%, 90 seconds, 77, 2 more seconds. Vote early, vote often. Okay. So, if we take a look at this, at your hypotheses here, we have 58% of you would hypothesize that this is an adaptation that has evolved 2 times within the deuterostomes that contain the echinoderms and the protostomes. So once there and once here. The other hypothesis that tied. Interesting. So some of you thought that, it arrived. So if you can, take your annotate tool and show me on the tree where, so for the 60% of you that thought that it had evolved 2 times, show me on the tree where that trait would have evolved to capture deuterostomes and protostomes. Show me like a heart or a star or something on which part of the phylogeny would then capture it. Yep. Now, pause for a second. I'm going to take a screen grab there, and I want you to consider, because I agree, if this is the hypothesis that if it's 2 times, it could have been directly in the branch to the echinoderms, which you could have also put it there and then presume that there would have been loss in the other deuterostome lineages. If it was here, this is 1 time, suggesting it's kind of missing the xenocoelomorpha, which are bilaterians, but it's saying, well, once and then it's been lost a bunch of times. Now, when we're talking about this, or this, or this, so the Faronids are where we see it. So we see it. So the relationship, the placement of your trait on the tree there depends very intimately on who's going to be related to whom within the Lophophores, within the Lophophorata, and the relationship between the Lophophorata and the Trochozoa. The Trochozoa is the group that contains the annelids, the mollusks, and the nemerteans. So there isn't actually a single answer, but I want to capture this. But I have a bit of a resolution here in a second. And I will clear. Thank you. And scroll this back up. So we can see within the echinoderms, so we've got placement here within Faronids, potential loss, and the fact then, there's clearly loss that's happening. And I think this is one of the things that I wanted to call attention to here, is that when I say that it's occurred within the echinoderms, it's not within all groups within the echinoderms. Some have it, half of them do, half of them don't. Many families, but not all of polychaetes have it. 2 classes, 2 groups of mollusks have it, some don't. Nemerteans have it, Faronids have it. So I guess just another worked example here of how we can look at traits and map them on a phylogeny and ask about consequences of different phylogenetic hypotheses, as one might do in an exam. So they secrete these chitinous tubes, and they incorporate sediment from the particles from the sediment into, so they have a nice tight tube. They never leave the tube. It's closed on that bulb on the posterior end. The swollen posterior is called the ampulla. So if you are skating this weekend and it's slippery and you have a swollen posterior, you can point at it and refer to it as your ampulla. And the lophophore can be automized, which essentially means that if it gets nibbled off, it can be regenerated. There's another close look at the photograph that we saw earlier on. They have what's called an Actinotroplarva, which is free-living and long-living, so analogous to the siphonaut larva that we saw within the Bryozoan. They don't have a loss. They don't have chaetae, which is a comparison to the Brachiopods, which we haven't arrived at yet, but which we will do very shortly. You can see the lophophore there. I'm going to jump through that actinotroph because I want to talk about the Brachiopods. 1009. So Brachiopods, this is a group that has, at some eras in geological history, this is a lophophorate-bearing taxa that has dominated our understanding of the diversity of life, particularly up until the Permian mass extinction event. They're solitary. They're benthic, they're marine, they still exist. There's not a whole bunch of species, just under 400 or 500. often living very deep waters. They're usually attached, they're a shelled organism, so they're a lophophorate-bearing taxa. We've seen that with the Bryozoans and the Faronids, where the lophophore extends out into the water. But in this case, if you're going to picture a brachiopod, if you haven't already, what you're going to picture is something like this in the bottom right-hand side. Some people called them in the 20th century these lamp shells because they resembled ancient Roman lamps. Brachiopod means arm-footed, brach, arm, pod, foot, and that refers to their peduncle, which is one part of the organism that stalks it and keeps it onto the substrate, and that we can eat in some cases. So one particular adaptation for the brachiopods here, this lophophore, this crown of hollow ciliated tentacles, is contained inside a shell. And a shell that looks externally very, very similar to a mollusk shell, to a bivalve shell. So they have no distinct head, and they have their lophophore. They're contained inside, protectively, inside shells. Now, where do they live? I think there is another copy of this video that will work. But marine benthic suspension feeder, they live just as many mollusks do attached to rocks. They're about 5 centimeters, so that's kind of large. It's like an order of magnitude larger than the Faronids that we saw. So it's a relatively large lophophore, and you can see here in this image, if you peer carefully, that frilly material, that's the lophophore contained within this larger shell. Now the shell is superficially, but like bivalve mollusks, but developmentally very, very different. So in this case, the shells of a bivalve mollusk, which we'll come to in about 2 weeks' time, or in a week after next, they're left and right. So the valves, the shell valves, the top and the bottom of that shell, are derived from the body on a left and right axis. The brachiopod is front and back, is a dorsal and ventral arrangement. So they look fairly similar. You can see just two shells, but I've oriented these pictures so that you can see that the plane of symmetry in bivalves runs right along the hinge, whereas here the plane of symmetry in brachiopods is perpendicular to the hinge. Now there's that video that is working, and so this is an older remotely operated vehicle that seems to be pogo sticking around on the bottom on the deep waters off of New Zealand. And all of these shells that are gathered together, boom, those are all brachiopods. And at times throughout the history of life on Earth, so here we have the Cambrian period, the explosion of life, the Ordovician, which is where the shells, the bits of rock that my parents took the picture of for you to explore, the Silurian, where Guelph was. You can see right up to the Permian that there were tens of thousands, many thousands of species of brachiopods that dominated life on the planet. And then whatever happened at the end of the Permian, it's constricted their diversity, or it reduced, it culled their diversity, and perhaps coincidentally with this, one might hypothesize that the diversity of bivalve mollusks radiated and filled that niche space. You can think about how you might test or examine those kinds of that kind of phylogenetic hypotheses. There's a particular species of a genus rather of brachiopod called Lingula, which is still extant today, and its evolutionary history, we believe, extends back 450 million years ago to the Ordovician. So we have these bivalve appearing shell and mantle. They have a calcareous support for the lophophore, because we're going to still try and support it within that shelled environment, but it has these brachial valves, and those arrange and essentially take what would otherwise, if you picture the lophophore in this case inside of that shell being drapes, and if you don't hang your drapes, they just lie in a clump on the floor and they don't do their job. And same with the lophophore, that it needs to be hung and extended, and that's what the brachia do. They have different muscle groups, so they have an anterior wall there. Now we're going to take a look at here. You can see the heart, that it's dorsally arranged. They have these adductor muscles. So the adductor muscle, the contraction of that adductor muscle pulls together the shell and it closes the shell. The diductor muscle here, when it fires, it opens the shell. Now, why would you? That's going to be a very important contracting muscular systems in this case, because to protect, one would presume that the valves provide protection from nibbling predators to the nice, fleshy, delicious lophophore. But as soon as you close the mantle, close the valves, you're breaking off the water currents that the lophophore needs to generate in order to generate food. Now the pedicel, which is right there, this is what's connecting the body. It's an extension of the body wall, and it's what connects it to the substrate. So there's another look, just before we get to the pedicel, another look from another text at the firing of the adductor muscle that closes the shell, and now below it the firing of the diductor that opens the shell. So another close look at this is the figure that we saw earlier with the frilly drapes of the lophophore arranged within the protective valve of the shell. And this is Lingula. This is the long pedicel that connects it to the substrate, and in this case is something that we harvest in some parts of the world and eat. So, Brachiopod morphology. We have ventral and dorsally arranged valves. There are two different kind of lineages within the brachiopods that are called the inarticulata and the articulata, and that refers to the presence or absence of teeth on the hinge and sockets. Most of the ones that are extant today are articulate, and so that has a hinge that holds two valves together in a more developed way. Most of the extinct lineages were from the inarticulata. Now they can actually, they're sessile and benthic, but they can move, I suppose in the same way, again, a callback to some of the movement in relation to predominant currents that we saw within other filter feeders like the cnidarians or sessile polypoid cnidarians, where we see burrowing and scissor-like motions within in the top that helps the brachiopod move itself and protect itself in the substrate, but then reorienting, perhaps once it's kind of moved and scissored down into the substrate, re-anchoring the pedicel, and then changing the direction of the shell in relation to the water current so that it's perpendicular to water flow, so that the current itself aids in the ciliary action of that the lophophore does to create a current. Think of an analogous situation to what we saw with the sponges, and that zombie experiment with the sponges, where currents were created both by the orientation of the animal and the structure of the individual cells, as well as by the ciliary action of the beating the flagella bearing cells within the sponge. So with the Faronids, one of the last things that we heard about was that they were closely related to brachiopods, but they didn't have small bristles or chedae like the brachiopods do. So this is my point to say that brachiopods have small bristles and chedae that extend on this mantle, edge here, and that we presume are using as tactile sensory devices. They also have chaetal sacs within the larva, which is what you're looking at in the kind of acorn shape in the bottom left-hand side, and then you're zooming in to an individual chedae there in that image, the rectangular image. So this is a soft tissue, and remember, most of the species of brachiopod described are fossils, and so we don't actually know directly about the soft tissue from fossils. What's preserved and transformed into the rock is, of course, hard tissue. And so in this case, we see in fossil brachiopods that there are grooves, similar grooves that we see, and so we think that they had the same kind of system. And this is a shout out to next week when we talk about annelids. This is probably an independent derivation of chedae to what we see with the annelids. So the lophophore extension extended on the brachias, you can see here, we're setting up very complicated inhalant and exhalant channels. So the brachium rising up and holding up that lophophore, and then an inhalant canal, so new water coming in, and an exhalant canal, so the water where the food has already been extracted through different parts of that of the lophophore tentacles. And so we see water flow and particle transport kind of separated to increase the efficiency of that. And it is about 20 past now. I'm going to put a little post-lecture, maybe 5 minutes, so I just wrap up the ends of the Brachiopods. I'm not going to keep us here longer because everybody's got places to go. We're going to rejoin on Monday, and if over the weekend you see this video, you might see a little 5-minute kind of, as I said, extension, completion of the Brachiopods. We'll rejoin on Monday, and we will then have moved to one of the most diverse groups we've seen so far, the annelids, so the truly segmented worms. So do something for your self-care this weekend, for your brain, for your body. Do something physical, do something that makes you smile, change your internal chemistry, and take care of yourself and take care of each other. And we will see you on Monday. I'm going to post links to all these videos and primary literature in the chat, and I'll be in the chat as I make the PDF and make the video to put on YouTube. So if you've got some questions, I'll go there now and I'll try and answer them. So have a great weekend. And goodbye.